So this has already been a very exciting day. Started at 5:45 this morning with the announcement that Victor Ambrose and Gary Ruvkin, longtime NIGMS grantees, had won the Nobel Prize in physiology and medicine. So we're very excited about that. But here we have another excitement, which is the Judith Greenberg Early Career Investigator Lecture. And this lecture honors NIGMS's former deputy director, Judith Greenberg, who was the longtime division director for the Genetics and Developmental Biology Division, or so named at the time. She became the deputy director of the Institute and was almost literally, my right hand for many years. And she was also the twice acting director of NIGMS. She was unable to be here with us today, but I believe she is watching on videocast, and we send her our warmest regards, and we miss her greatly. As we begin the lecture, please note that it will be recorded, and so the proceedings are being recorded. We launched the lecture in 2016 to showcase NIGMS-funded early career investigators and the many very valuable contributions they make to biomedical research. We gear the lectures to students to give them an opportunity to hear and meet with early career scientists who are doing many remarkable things. And our hope is that in so doing, the students will be inspired to pursue careers in biomedical research. The lecture is going to begin with a speaker, Dr. Saad Bhamla, giving a 30-minute talk on his research and career path, and then that's going to be followed by about a 30-minute or so question and answer session. And I encourage all of you to ask questions of Dr. Bhamla about his research and his research career path. You can either submit them to me through the Zoom chat box, or if you are here in the room, and we have a wonderfully large crowd in the room today, you can come up to the microphone to ask the questions. We've also gotten a few questions through the registration form already, and we'll be trying to ask those as we go along. So today's speaker, Dr. Saad Bhamla, is an associate professor in the School of Chemical and Biomolecular Engineering at Georgia Institute of Technology in Atlanta, Georgia. His lab explores fundamental and applied research questions through the development of new experimental tools and techniques at the intersection of soft matter, organismic physics, and global health. Saad studies how single-celled organisms called ciliates use protein engines powered by calcium to achieve ultrafast motion. He's uncovering the mechanics of these calcium motors, which differ from ATP-driven motors that we're more familiar with, that are found in most forms of life. He's also at the forefront of an emerging field called frugal science that works to develop affordable and accessible tools for global health research applications. Dedicated to making science accessible and enjoyable for everyone, he also creates multilingual comics about his research, and I happen to have a stack of them here. And so those of you in the room who ask questions may be given a comic as a reward for your curiosity. Recently, Saad won the prestigious Moore Inventor Fellowship from the Gordon and Betty Moore Foundation, as well as the Young Faculty Award from the Defense Advanced Research Projects Agency, DARPA. NIGMS is very proud to fund Saad's research and outreach work through both our R35 Maximizing Investigators Research Award and through the Science Education Partnership Award. Saad, welcome, and we look forward to hearing your talk. Thank you.
Thank you. Really great to be here. And I'm going to tell you a few stories once we have the slides up. Okay, all right. So I'm going to tell you a few stories. Our lab is essentially curiosity-driven lab. It's a bunch of physicists, engineers, biologists, roboticists, and we look at living systems because we believe that we look at them as nature's engineers that have solved some of these puzzles, and we try to kind of get these kind of deeper truths through them, bringing kind of these lenses of different fields. In this spirit, we're very promiscuous kind of in the systems we study across scales, all the way from single cells to organisms to collectives of them. Today's talk is, of course, going to be on some of these calcium-powered cells, as John alluded to, but we've thought about why do we blink? So as you're looking at it, we looked at mud skippers to understand the evolution of blinking, to the whimsical as to how insects pee and why do they pee, to thinking about worms that may tangle up and create topological active matter, as well as teams of dogs in Alaska that may work together and how they perform tasks. So today's story begins a few years ago, where a group of us were thinking about some of the fastest movements in nature. And on this y-axis over here, I'm showing you acceleration. It's on a log scale, and on the x-axis is essentially a length scale. And what we started to appreciate is that in nature, there are many organisms that achieve accelerations that are just at really small scales. And they beat some of the engineered systems, such as like tiny robots or these soft robots, which haven't passed this millimeter length scale. So nature is able to achieve really, really fast accelerations. And in fact, there are these open spaces over here, especially in the micron region, that I was both excited to see, because it kind of meant that there is a lot of scope for discovery. And this kind of upper limit on the top right, that's a nematocyst from a jellyfish that can accelerate when it shoots out a harpoon at nearly million g-forces in nanoseconds. But there is no speed of light kind of limit to acceleration. So there's lots of room for discovery on what organisms might kind of break that. And so here, as I was starting out my faculty career, I was excited to kind of explore what kind of organisms could fill up the space and how they may achieve this, especially at the single cell level. And second, how could we harness some of these principles to do some engineering, especially, say, in synthetic biology? As I was kind of exploring some of these single cell systems, I kind of appreciated that what they lack in size, they make up in numbers. There's just this immense diversity on the planet for single cells. They come in all shapes, colors, and forms. And one in particular kind of captivated me. So let me introduce you to the protagonist of today's story, Spirostomum ambiguum. And the first time I lay my eyes on Spirostomum through a microscope, I was just, blown away by its immense size. It's one of the world's largest cells. It can grow up to 4 millimeters in length. You can see it with the naked eye when we hold up cultures. In fact, you can see as it's swimming through, it's bumping some of the other research organisms like Tetrahymena as if they're nothing. I think of them as the dinosaurs of the microbial world. And what really got my attention is, as I was watching it, I thought I had blinked, and perhaps you will too. You'll see it does a very peculiar behavior. So it's a little bit laggy on Zoom, but it really can contract really rapidly. And it does so spontaneously. So for example, in a culture under a microscope, these cells are going off as fireworks. They're spontaneously contracting as if there is a purpose to this. And so that was kind of the first puzzle I wanted to solve. How are these cells contracting so quickly, and why are they doing so? The why part turned out to be a little bit easier to solve. And this is a fluid dynamics trick that we applied, which is to take these cells, confine them between a glass slide and a cover slip, and throw in some plastic particles. And what that allows us to do is to trace out the fluid vortices, these bipolar vortices, these really beautiful and gorgeous vortices that they make as they contract. And this kind of is one of the first discoveries and aha insights as to why they may be doing it, is because typically we think of single cells as confined to low Reynolds numbers, but these, when they achieve this contraction, you can see these long-range vortices go a few body lengths away. And the Reynolds number over here, the transient Reynolds number, is about 100. And so they're doing this through this behavior. And as we discovered in the story, is that they're doing to talk to each other. We call this a new form of communication, which is purely through mechanics, purely through hydrodynamics, where it's a chain reaction where one cell kind of contracts, sets up a wave, and the others are they're basically the integration of sensing and actuation is coupled into these cigar or antenna-shaped living systems, and they communicate over long distances in colonies of hundreds of these cells. And so this was kind of a first aha moment for why these organisms might have evolved these ultra-fast contractions, so that they can kind of beat the Reynolds number to be able to set up these long vortices so that they can communicate about each other. The second part, where we've spent some time thinking about how they actually generate these forces and what is the machinery that allows these cells to contract. Over here is a fluorescent labeled confocal image of a spirostomum in its contracted state with a tag for centrin. And really what we found out is that the contraction phase does not require ATP. When they re-extend is when you require ATP, but this is a calcium-driven engine, and it forms a fishnet structure. And in fact, you can see a slow motion of both the cell and a model that we built that reveals a calcium wave essentially going down the length of the cell as it communicates over this giant cell, a few millimeters, and it synchronizes this spring or myoneem engine. In fact, if you take some of these numbers, and this is what in the battery community people conventionally do, is to plot the specific power as a function of specific energy. Essentially, that tells you, for example, you want a battery that has a long power capacity. You want your phone to last longer, but you also want the charging to be fast. So the power is essentially how fast it can discharge that, and specific energy is how much can it store. We find that if we compare it to the different engines that we know across living systems, compared to the venous flytrap, our own muscles, the myoneems compared to a two-stroke diesel engine. There's a lot of power packed into these calcium-based springs. They're one of the most powerful cellular machineries we know. And so this kind of led to thinking about how would we kind of harness this. And we're developing a paper now where we're trying to understand what are these myoneems composed of. And to the best of our understanding, they're essentially a composition of these two giant proteins. One is centrin, which is the calcium responsive part, and it's decorated on a backbone called SFIO1, and it forms this repeat. So you essentially have calcium waves going through as a calcium wave releases, and then you have this kinking of the backbone to create this motion. Of course, these are at the nanometer scale, so you essentially have a hierarchy of the structure occurring across multiple scales to give the motion that we saw on a millimeter scale, and that's something we still don't understand, and we're kind of working towards to understand how these nanometer scale protein, supramolecular protein motifs actually lead to motion at millimeter scales. And this protein assembly is actually conserved across different ciliates. So I'm highlighting two. One is tetrahymena and then spirostomum. And there are different kind of homologs to these calcium binding proteins. The 25 kilodalton example is TCB2, which our collaborator, Jerry Hans, who's at Drake University, essentially focused on for his PhD thesis. And over the last few years working with him, we've been able to express at least the TCB2 protein, these calcium-triggered proteins in E. coli, such that we can start to play. So we're taking this top-down approach where we're watching nature, and as the Yogi Berra quote goes, we can watch a lot by learning. And then we're kind of putting a second hat where we're trying to do things from the bottom up, kind of channeling the Feynman approach, is like, what I cannot build, I don't understand. And so this is the second approach where when I saw this video for the first time, this is, again, just this calcium responsive protein and a calcium wave, calcium droplet using a pipette on the right-hand side. You can start to see this protein self-assemble just because of calcium. In fact, it's starting to self-assemble into a network, and you can start to see emergent of these waves, which indicate that it can already generate forces. Now, this is remarkable to me when I saw this, because it tells you that there's a whole community that thinks about actomyosin systems and reconstituting these milieu of cytoskeletal assemblies that require so many proteins. And here we have just one protein and calcium, and you can generate forces and motion. So it's a much simpler constituent if you want to animate life in synthetic form. And so one of the challenges with the calcium is that you can't control it in space on time. And so working with a phenomenal graduate student in the lab, we essentially built a projector, but for a microscope. It's known as a digital mirror device. And we used a clever insight is that we can cage the calcium using this DMNP-EDTA. Essentially, this is a giant molecule that gives calcium a hug. And as soon as we shine light, it releases it. So calcium is present, but it's protected by this molecule. And so if we shine a light, say in the form of this shape, on the right-hand side, you can see the network form, but now in the shape. So now we can controllably self-assemble this network. But the beauty of the system is we don't have to do it in just one go. We can start to use the shading or the gradients or in time, so we can start to move patterns around and start to self-assemble the motion, self-assemble this networks so that we can generate force wherever we need it. And so I'll show you a few insights as to what we've been able to do with this. On the top shows, for example, one image where the protein has assembled. It's a DIC image, so you can see the gray part is where the assembly has taken part. And on the right-hand side shows you the same video we saw as a series of images. And we wanted to understand, although it's a simple protein, which is DMNP and calcium, to model this required these sets of equations. But what this leads to is this beautiful insight. And this is one of my favorite images where you can see the simulations match the experiment so well. We're able to, because it's simple, be able to develop a chemomechanical model that allows us to understand what's happening, both in terms of the concentration of these proteins, but also where calcium is in terms of its concentration. And we can track all of the different intermediate proteins in this network. This helps us develop some control over this. And we made a simple discovery is that in this light, if we actually pulse this light, we can actually age this network and grow. And this to me looks like a small protein heart because as you pulse this, you can start to see contractile patterns emerge in it. And the PIV, which is essentially the force analysis on the right-hand side, shows you that as you pulse this network, it grows stronger and it exerts more forces. In fact, it doesn't just exert inward forces. We realize that this system actually reveals beautiful divergent forces. So you can both push because it creates pushing forces, but also pulling forces based on the radial direction where it is. And so we leverage this insight just to give you a feel of where we're going for. Here's an image where we're going to pulse this light, and there are a bunch of droplets, particles, and synthetic vesicles. And you can start to see we can push them away, but also we can pull them inside. And so essentially in this network, by using this light control, we can displace and push particles. And the vision we are thinking about is inside a synthetic cell, you could position or place organelles. And so these protein networks generate enough forces to achieve animation or locomotion of particles. And here's an example trace of pushing a particle as well as pulling a liposome. Here, of course, I have a talented grad student who was doing this. Recently, we've been trying to develop basically a two simple network level reinforcement learning pipelines such that we can tell the code where we would like the placement of this particular particle, just so that a human doesn't need to involve, and you can use the chemomechanical chemistry of this thing to direct where this particle might be. And so this is some early work as we're bringing in new tools to study this. I'm going to transition to telling you a little bit about so this was my MIRA grant, and then I'll tell you a little bit about my SEPA grant from NIGMS. I feel like I have two hats on, and hopefully by the end of the lecture, it'll become clear how they're connected. I had just finished my PhD from Stanford in 2016, and I had the opportunity to be in Madagascar. This road that you see over here is in Rana Mofana. It leads to a village that we hiked on foot for about 3 hours. And it's a village, serves 500 to 600 people. And I met Raisa, who's their only community health center worker over there. And she showed me in this small hut that serves as the local medical facility, the box she has, which has a few lancets, a few rapid diagnostic tests, and paracetamol. And essentially, I kind of appreciated the challenge for a remote place like this, but Madagascar, where if there is an emergency, you have to, walk or on a motorbike, and you have to cross a river if there's a medical emergency, because this is very, very limited. And I saw the local village school and kind of broke my heart. And this was kind of a transition moment for me, where I realized that I was going to dedicate the next part of my career with 50% of my time doing basic research. But there's an engineer in me that wanted to think about ways that I could help both the challenges in global health, but also education. I had grown up in India, and in 2001, these are my grandparents. My grandmother was losing her hearing, and I had just gotten into undergraduate college, and I thought, wouldn't it be cool if I could buy hearing aids for my grandmother? And it was very sobering to realize that I could afford a phone, but hearing aids cost $3,000 equivalent, and I could afford a phone, but not hearing aids. And that kind of struck me that hardware, which is both an enabler and can allow us to live our lives much better, it's also an impediment because it can require resources, electricity, cost can be a huge barrier. And this kind of sets the philosophy for how we approach developing hardware. We call it frugal science, with the mission of democratizing access to scientific hardware, such that we can do diagnostics under the tree, we can do science deep in the Amazon rainforest, or even in my own backyard at a local public school in Georgia or Alabama, where I live, because these science labs are very, very resource constrained. The frugal science philosophy is a little bit divergent from how in Western science we think about how we do science. It's essentially Western science goes about thinking about technology as asking, what would Croesus do? Croesus was one of the richest kings in Lydia. He was so rich, he was the first to issue gold coins. And essentially we ask ourselves, what would we do if we had unlimited resources? Whether it's sequencing the human genome, creating a human cell atlas, or mapping the human brain. I take a different approach. I ask, well, what would Gandhi do? I put costs front and center, and we put ourselves around a box of a few dollars, and then we think inside this box to come up with creative solutions to some of these challenges. And we do this because we share this finite planet of ours with billions of people that live on a couple of dollars a day. And so it's important to be thinking about costs as we think about these devices. I'm going to tell you 3 inventions that we've developed over the last few years. The first is a hearing aid. So this is to address the challenge of hearing aids in low and middle income countries, just like my own grandmother. So I built a hearing aid that costs 98 cents. And we've shown that it's as effective as commercial hearing aids. And currently we're working with partners. We just finished a pilot trial in Malawi. And we are working to scale it up to 10,000 units, both with local addressing kind of the logistical challenge of not just importing devices, but actually developing local economy to construct and manufacture these locally. The second device is essentially a centrifuge. It's, again, one of those workhorses. I think everyone in this audience has used a centrifuge, either in any molecular biology lab or for diagnostics. But these things cost thousands of dollars. And so I came up with the idea to invent one just using a children's toy. And this is just a piece of paper, strings, and dowels that I bought from Home Depot. It's a 20-cent paper centrifuge. And what's remarkable is it goes at 125,000 RPM or 30,000 G-forces. And this is really useful because I could take a capillary and I could take a droplet of blood, about 10 microliters, and I could stick it into my paperfuge, and I would spin it for about 30 seconds to a minute, and you would get instantly a simple test for hematocrit analysis that would help you diagnose for anemia, which affects billions of people. In our paper, we've also shown the same could be applied using a slightly sophisticated technique called quantitative Buffy code, and you could do malaria diagnostics, filariasis. And so it's just a powerful platform tool. And finally, all of us today have access to this modern miracle mRNA vaccines that help us keep healthy. But even today, billions of people don't have access to mRNA vaccines because of the challenges of cold chain. And there are other ways to deliver mRNA vaccines that don't require lipid nanoparticles. And those alternatives, a physical way rather than a chemical way, are known as electroporators. But these things, again, same old story. They're bulky, cost a lot, and just not designed for low and middle income countries or point of care. And so I had the insight to develop this. I was doing a barbecue, and all of you have seen this device, use this at home. It's a barbecue lighter, good for thousands of clicks. And then, of course, we make them by the billions each year. And the aha moment was that every time we clicked, we generated a spark that, of course, would lead to a fire. But this spark could be used to create nanopores, essentially use piezoelectricity to transfect living systems. We showed that this was an effective way to do both E. coli, but lately we've partnered with colleagues to put microneedles on top, and we've developed a $1 e-patch, which we showed in recent papers that for SARS-CoV-2 provides strong immune response and is also very, very tolerable and safe in humans. We've spun out a company now, and it's got 6 full-time employees. And essentially, we're very impatient optimists, and we're not waiting for other people. We're going out with the goal of bringing mRNA vaccines and all the other flavors of RNA-based therapeutics to from a global perspective. And we're onwards working with pharma companies as well as thinking about the FDA submission. I want to tell you a final story. When I showed Raisa my paperfuge, because I was training her how to do this malaria diagnostics, she laughed at me. And she said, "This is the fancy centrifuge you came up with your fancy degree at Stanford." And then the next day, she had taken the jar from a pickle jar, the lid from a pickle jar, and she had put 2 holes in it and taken some thread, and she had made her own centrifuge. And she said, "Saad, look, I'm an inventor too." And I think this story gets to the heart of frugal science. It's not just about building accessible, scalable, and affordable tools. It's democratizing kind of the joy of discovery, the joy of invention, is that anybody, anywhere can solve problems and invent tools. And so to channel this, we've been lately thinking about how, we used to be in an era where we would think all children should learn how to code because the era was going to be a digital era. And we've shifted now, and testament to today's Nobel Prizes, that we're moving into an era of thinking about coding life itself, understanding how the molecular code of life and how do we manipulate it. And so we have this vision that we have our teachers and students, high school students, who we want not to just learn how to code software, but wetware. But there's a huge challenge between coding on a computer versus coding and doing, whether you're doing CRISPR or gene therapeutics or just even plasmid engineering. It requires immense resources. And how do we bridge that gap? This is an example from about 14 years ago of the cost required to do synthetic biology or molecular biology. Each one of these instruments is kind of on my hit list, and one by one, we're going to invent a frugal version of it such that we have a lab in a backpack, such that people can do hands-on STEM when we say it should be hands-on STEM, and not just watch a video about how CRISPR should be done. And so with this, we've been with the SEPA grant, we've been essentially training teachers onto doing hands-on STEM biology, whether it's from Thailand to rural Georgia. In fact, these 2 inventions are from our high school students, part of the Frugal Science Academy, where they develop new tools to extract DNA as well as track living things. And so essentially, so far, our impact have been that we've, over the last 2 years, trained 45 teachers. Each teacher across different school districts has an amplification because they incorporate some of these ideas into their curriculum. And so it reaches many, many students. And as I showed you, we've also, thinking about translation of these devices. I thought I'd end on essentially, don't take my word for it. So I have 2 teachers who wanted to kind of communicate to you what this means to them. So we'll have 2 kind of teachers tell you what the Frugal Science Academy means.
Hi, my name is Melissa Rowe, and I'm a science teacher at Chattooga High School in Summerville, Georgia. I teach chemistry and physical science to 125 students in this rural Northwest Georgia town. Prior to Frugal Science Academy, I had little to no knowledge of synthetic biology and biotechnology. Through this training, I have become very confident in teaching my students about techniques, skills, and knowledge of synthetic biology and biotechnology, such as gel electrophoresis and PCR.
For the past couple of years, I've had the opportunity of attending Frugal Science Academy workshops with a wonderful cohort of teachers that have been essential for enhancing my teaching skills, strategies, and providing me with the resources that enables me to bring biotechnology into the classroom. I work in a Title I school where students were able to perform genuine laboratory experiments, and our newly formed BioBuilders team was able not only to develop a project addressing real-life problems, but they were also able to present their project at MIT in Boston, which was an invaluable opportunity for them in which I would like to continue. Frugal Science Academy, with the support of NIH, has given me the confidence and support to successfully design a program for my school. Thank you.
The COVID pandemic has shown us that even though we build kind of frugal inventions, it may be simple cloth masks or vaccine, without an educated and trained workforce who believes in science and who believes in the power of science and how it works, we're not going to be able to take these life-saving technologies and be able to, empower people to use these things to save their lives. And which is why I balance my time between actually doing the science, but also training the next generation, whether it's in Thailand or in rural Georgia, because I think we need to think about the next generation and whether they, how they understand and practice these technologies that will help them and us address some of the planetary scale challenges that we're left with. With this, I'll thank you for your attention. Really grateful to be here. And as John mentioned, I brought some of these comics over here. That's my wife reading to my 4-year-old. So it's been validated and proofed on a 4-year-old, and it's engaging. So thank you again for your time.
Thank you, Saad. That was fantastic. Very inspiring talk. So now we have the question and answer session, and we do have some questions already from some students out there, but we invite you as well in the audience to come down and ask questions and perhaps receive a prize. So one of the questions, Saad, is how did you get interested in science in the first place, and how did you get going in your career?
It's actually, I've had this discussion with reporters who want an origin story in which I wish I could say, oh, I've taken apart radios and got bugs when I was a child, and I knew. I was actually a failure. I'll tell you a story. I got 0 in math one day, and I went to my dad, and I was very scared because he had to sign it. And he laughed at me, and he said, oh, this is great news, and we should laminate this. And I was a little bit confused. And I was like, wow, I don't understand what you're seeing in this. And he says, well, this is great. 0 is good because this is rock bottom. You can't go any lower than this. There's only one way to go from here, and it's going to be up. And it's kind of that attitude that helped me. And I feel almost like an imposter, the kind of fields I work in, whether it's working with DARPA to improve marine vehicles, or playing with worm blobs in my lab, or developing hearing aids. And I think I just trying to figure myself out and recognizing it's okay to, have kind of an off-beaten path is okay. So I give myself permission to do this. And I essentially, whenever I find students who don't know what they want to do, which happens quite often, I think that's okay. I feel very grateful to be able to play with bugs in the Amazon as an adult. And I'm so grateful I didn't do it as a child, because now I basically can make a living out of it.
So you mentioned you feel like an imposter. Can you explore that a little more for us? Because I think many people, young people especially, have that feeling.
Yeah. To me, I don't mean it in a negative, debilitating way. I actually mean that I'm very insecure, but I think in a very positive way. It actually helps me be I'm positively insecure. As I know, most artists, most inventors, and most creative people are. It helps me keep pushing to understand and learn new things, because as we all learn, we realize how much we don't know. But it doesn't stop me. It actually keeps me just going. So I feel like an imposter. And the day I feel like I know too much, I know I'm probably something is off. And so I enjoy not knowing, and I enjoy feeling stupid while doing research, because I feel like I'm pushing the boundaries and asking, not just answering, but asking interesting questions, for me at least.
That's great. What are some unexpected places that you found inspiration for a device?
So the paperfuge idea was when we were not very well off when we were kids. And so my grandmother, to keep me entertained, used to give me buttons and strings. And so I used to play with that. So I had this thing from my childhood that I was playing. And I learned that many cultures have this. People use either Pepsi or Coca-Cola, the metal cap, and make holes in it, buttons. In fact, it's one of the oldest toys. It's called run run or whatever in your culture or language. And there are many archaeologists have found many tablets. They didn't know what it was. It had 2 holes. And then they realized, oh, the idea was it would ward off spirits because it makes a sound. So that's a fun one. The barbecue lighter was a good one. We've had, there's one we're working on, which we are very excited, and arguably, I think one of the most exciting projects. We're thinking of it as a universal diagnostic method, which is some of the support we got from Moore, which I can't share more about for reasons, but the inspiration for there was also kind of a very strange and interesting way that we came upon the aha moment to kind
We'll
of,
have to wait for that one.
yeah, we're working hard on it.
That's great. So one person would like to know if you can give some examples of end products, health interventions that have come from research in low and middle income countries.
I'm assuming not just from my lab.
Not just from your lab, but
From research in low and middle income countries.
that's what they're looking for, maybe some.
So the Jaipur foot is a phenomenal example of prosthetics that are locally made, and it's one of the classic examples where they basically make local custom prosthetics and have helped. I think they've been doing this for 30 years, and it's a role model for frugal innovation where it's not high tech, but it can help somebody become mobile and move. The other thing I know is there's an eye surgery. It's in Nepal or in the Himalayas somewhere where they've figured out it's almost like the Henry Ford model, but for eye surgery. And because some of the populations in these places, they've figured out a way to really trim costs and help people with cataract surgery, and have basically given address surgery for, I think, a few million people as they grow older, especially in high elevation. So I think there are just countless examples. Sometimes it's like tuberculosis. It's one of the deadliest diseases on the planet. Kills somebody every 20 seconds. But most people, at least in where we are, NIH, I'm sure people know, but in many people I interact with, I'll say tuberculosis, and they may not even know. And it's kind of this thing, we live in bubbles, and sometimes we may focus on something in our own backyards rather than.
Right. Couple of questions now that have come in on the chat. So why did evolution go with ATP instead of calcium, given calcium can do so much?
Oh gosh. I think really don't think my chemical engineering degree equips me to make claims about evolution. At least one perspective I have is that it's like a toolkit that evolution may have access to, or organisms may have access to, and calcium works for something and ATP may work for the other. It's not as if we don't use calcium. Like the fact that we have our hearts breathing is because of calcium. And in fact, centrin beats at the heart of every eukaryotic cell because it's actually involved in generating forces when cells divide. So it's just there. It may play a role that we may not quickly appreciate, but it's some of these proteins are
as much. So the second question is, do you get pushback from companies for making these cool and immensely useful pieces of equipment that sell for only a few dollars instead of thousands of dollars?
I wish, not yet. I think if I'm being very critical, one of the challenges with some of these technologies is actually translation. And it's not just for this technology. Anybody who invents anything, we all know that there's one thing to write a paper, another to patent, and you can even license it, but there's a whole other thing to take it to market and translate. And there's a lot of challenges with just that. Take the hearing aids, for example. It's the same old story. The engineer in me, I've changed my approach, but as a technologist, it's very easy to think technology is the answer, that if I build a hearing aid, everybody will wear one. And it's much more complicated than that, because as we go do our field work in India and Malawi, you can offer a hearing aid to somebody who is actually aware that they have a hearing loss. What do you do with that? And so you can't just ship hearing aids. So there has to be some. So now we're thinking about, well, how am I going to get this hearing aid to a farmer who is, is so far away from a traditional distribution source? So it's these additional challenges of social education, distribution, logistics, local manufacturing, and education, not just at a personal level, but at a policy level, at a governmental level, that it's very I have this funder who, when I had the idea for a $1 hearing aid, I wrote to them and they gave me my first $10,000 grant. I was a young, naive assistant professor. And I was like, yes, $10,000. That's amazing. It didn't last that much, but it was great. But then, of course, that was 7 years ago. So I had to go back to him saying, well, we have more stuff to do. And he was like, yeah, I'm happily in this is Capita Foundation. So Robert Capita, he gave me another check and another check and another check. And he kept telling me that we have to keep putting the drops in because the difficult things we do right away. The impossible takes a little longer. And he appreciated that the technology part, as he could see, and I didn't at that time, that we would write this paper and everybody would have hearing aids. It doesn't work that way, which is why we started a company as well. And just the long haul, it's just the long game.
Yeah. I invite people to come down and ask questions as well. There's a mic here. Along the same lines as, whether companies feel threatened by you, is there a concern that making synthetic biology tools too accessible could have, dangerous implications with people making weapons and things like that?
I think it's just like we do this with every technology, whether it's ChatGPT, our kids gonna, copy, or with coding, are we gonna have hacking? And the goal is to not, put our heads in the sand, but to really start. And so one of the things this year we're starting with the CEPA program is including ethics, because nobody teaches ethics to high school teachers and high school students. But we are trying to teach them how to do CRISPR. They did CRISPR this summer. Well, okay, let's talk about the ethics about this. And so we're creating curriculum for this, because some of the ethics curriculums are from 2010, but that's already 14 years too old. Like, things move so fast. So I think the important thing is just to think and figure this out together and address this and not just ignore it, because today you can put a code and you can get a plasmid, and you could get a plasmid for a novel flu strain. And I don't think many of the companies that are shipping DNA, RNA are even checking. So I think the US as a whole should be aware of how it's not a high school student, but it could be, scientists who could create trouble. But I think at a more policy level, we need to be aware of the challenges of these things.
Teaching it early is a great idea. Dorothy?
I have a question for each part of your talk. One is about your bottom-up approach to looking at the calcium-mediated contraction. Is that with just a single pure protein from your colleague? So that in looking at your images, my question was, what do you know about the assemblies that form from this pure protein in response to calcium?
So we're just actually, as of last month, doing cryo-EM and trying to understand how they self-assemble and what is the structure that they self-assemble into. We don't have the data right away, but that's essentially the ongoing work. But right now, that just one protein and calcium.
It's amazing.
It's remarkable. And on parallel thrust, as much as we are skeptical, we are trying, which is using AlphaFold and other tools, just to give us some understanding of, well, how is it conforming? How is calcium binding? So we're trying to approach it, but we're working with, we have a grant from NSF with 5 PIs. And so I'm very grateful, because I thought I was only obsessed with this organism. It turns out you can kind of make other people both first fascinated and then obsessed. And then you're like, okay, we got to solve this puzzle together. We need more men and women, more smarter people to think about this. So it's a great team, and we're approaching it from so many different angles, and also expressing kind of the backbone protein, because this is not enough yet. I would like to.
Yeah, you want to be. Yeah.
So we want these squishy cells, because when people say synthetic cells, it's like the minimal cells. You want to make a minimal genome or something like that. To me, the hallmark of life is that it's animated. And I want to make animated cells, not just bags of enzymes or RNA. And so I envision we can make a synthetic amoeba cell that could crawl. So we need more force. So we need that backbone, because this force, it can move a liposome or a particle, but it's not going to deform some complex in a viscous goo. So we're working on it. But
Question about part 2 is whether, as you create these frugal devices and cost-effective devices, do you ever have any pushback in the sense of accepting it simply because the model thus far has been more expensive technologies. And even among low income and middle income countries, there may be a predilection to that the more expensive is better. So has that ever entered into the acceptance of your technologies?
That's what we thought when we did the Malawi pilot, and we had this old grandmother who was part of our thing, and we gave the hearing aid for a month. And she had a lot of issues. She hated that purple color. She had lots of opinions on everything, right? And because for the entire country of Malawi, they rely on refurbished hearing aids from UK. And after a month, she gave us a lot of good feedback, and it's all in Chechawi, so we have to translate it to English. We have translators. And then we said, well, was it any benefit? She's like, the only thing that helped me is every Sunday I could go to mass and I could go to church. And that's the time, that half an hour, 20 minutes on a Sunday, it was helpful to me. And so that was a positive feedback as an engineer who's shipping out things, because that takes a lot of, and so he said, okay, can you give the device back? And she said, no, it's $1, right? I'm going to keep it. And to me, that was valuable because she complained about it and she said she didn't like the color and everything, but she refused to give the device back. And I'll take that as a win, that we're doing something right. And for $1, the goal is not to build the frills and whistles of machine learning in your ear and Bluetooth. Is that what value could it bring to a person who has nothing? And can that help them for 20 minutes on a Sunday or have a meaningful interaction? And that's all we're looking for. It shouldn't replace everything. We all need different things. We need our Roombas, but we need a broom someday. It's just that same approach can we have. And so I think there is some pushback sometimes because there is a perception issue about cheap not necessarily being bad, not necessarily being good. But also when you have nothing, we see this because we do field work, there is hunger. That hard information is literally free. Billions of people have access to phones and can access information via Wikipedia. But all of us are scientists because at some point we had to do things hands-on. We did our first PCR gel. We did our first chemistry. The hands-on part, the joy of science is through instruments, whether it's a microscope or a telescope. And so that makes the difference for a kid or somebody who needs a biomedical device. And so at some point, if it works, they don't care the color and the cost. Yeah, but sometimes our reviewers for that paper for the centrifuge, the reviewer number 1 gets submitted to a nice journal. It came out nice, but he was like, I don't understand what this whole huzzah is about. 6 pages of a paper centrifuge. Why don't they just use electricity? So I think sometimes,
That's the point.
yeah, it was like very hard for them to rationalize. Like, just plug it in.
So
So it's very hard to battle.
before we go to more questions here, there are a few more.
Please. You've earned it. You won 2 because of your 2 questions.
Couple of questions online. So what's your advice, Saad, for PhD students interested in either working in your lab or in, this sort of field in general, both from the US and from abroad?
I think we just have to be ourselves and try to be the best versions of ourselves. There's always, I think I'm very fortunate. I'm at Georgia Tech and grateful for an IGMS, but also I could use that money to do, I think, traditional things, but I just kind of figure out what my inner voice is and do it. And so I think we can all worry about tenure and awards and stuff, but at some point we just have to do the things we want to do, because what good is all this money if we don't actually play and both do the serious stuff, but do the playful part too? I think a little bit is okay, a little bit whimsical, a little bit of idiosyncrasy is okay. And so people should just kind of find that space. And I've never had too much issues meeting PhD students, and oftentimes they all have their weekend, Friday afternoon projects. They all find their space. It's just that sometimes the nature science, the grants, the fellowships, that drives it. And I think it takes a little bit of effort, but if those 2 can match, as I think I've figured out, then it's fun every day. Yeah.
So certainly the mirrors, we're trying to create a little bit of space for that. And then the second question that was online was hearing loss can be different in different people.
Yeah.
Can you tune your device depending on the person's particular situation?
So not at the moment. We had to prioritize. We focus on age-related hearing loss, which has more of a sloping increase after 1 kilohertz of frequency. So we had to focus on the demographic that we could. Within that, we have a switch for mild versus moderate, similar to what the FDA OTC guidelines are for over-the-counter hearing aids, which is why you don't need audiologists. So right now, the goal, you have to cut somewhere. And
Right.
so.
Okay, we're gonna do it lightning rounds. We got 5 minutes. Go.
Okay. So you have research grant and also you have science education grant, SIPA program, SIPA. And SIPA is for K-12 science education. I think there are people in the audience who may also interested in science education, and some people may not know SIPA very well. So can you share more information about SIPA, your experience with SIPA, and also how you balance the research and science education?
Before SIPA, I was doing kind of the science education for free, meaning I was using my faculty startup. And then one faculty who had a SIPA grant saw my work and he's like, why don't you have a SIPA grant? Shouldn't you be using your startup? You should go to NIH and they have a program. And it's been amazing because we were able to scale from a few teachers to now 20 to 45 teachers. And the science education partnership grant is, I think, amazing. This like gives you 5 years, gives you a runway, and allows you to scale, because we're already starting to think what the next version would look like. And we scale up from Georgia to, perhaps national level, thinking about the curriculum. So for folks who don't know SIPA, I think it's one of those hidden gems. It just allows people to actually execute some idea that might be there, and yeah, do the meaningful high school education. You get a comic.
So I enjoyed your talk. My daughter got very excited about doing science with like a strawberry, the plastic bag, Dawn dishwashing liquid, and basically extracting DNA, right? So like that's like for her was like hands-on and being able to see something for real, and that costs like pennies. So love what you're doing. My question about your paper centrifuge is, how do you get consistency in that?
Yeah. So that's been some of the challenges in translating it, especially for, I was there pulling samples for malaria diagnostics in Madagascar. And that is one of the challenges we faced, is how do you actually train somebody and how do you make sure, well, now you stop because now you've gotten some of the things. And so there are limitations with such a device, and which is why we haven't spun a company out and it's become a translation device. What has happened is that there is this surprising similarity between the global health and the science education part. So it's been a win on the STEM side, because we built a 3D fuge and we use it for molecular biology, both in the Amazon, but also high schools. So it works in that sense much more reliably and beautifully, because if you're just extracting DNA or doing kind of molecular biology experiments, it works quite well. So that's the part we haven't challenged, but one of the things that happened is this beautiful explosion of kind of these devices. So people have made kind of thinking about the same thing. One of the innovations was that people prior to this had thought about only salad spinners and egg beaters. Essentially, the trick over there is you're using mechanical gears to convert muscle energy into rotational energy. The aha moment here was we use the super coiling of strings. In fact, this is hidden in the SI, about 20 pages of math to figure out the super coiling strings. It's borrowed from Zev Bryant's at Stanford, the DNA coiling experiments where they tether DNA. It's the same principle. So we use strings into springs to convert motion into rotational energy. And so there's been an explosion of devices inspired by this for charging up batteries and adding some of those kind of controls to make this a little bit more reliable and robust. And people have had better use for some of the urine-based tests where they want to extract things. So this may not have, but there are essentially, I think, about 50 or 60 of these kind of offshoots. So
Thank you.
Yeah.
And please, for you. Olga, last question.
Yes. I don't know whether I had chance to do it, but if you will mutate this new protein so it won't bind calcium anymore, how does that will affect behavior of these cells and whether they can divide after that or not? What happened to them?
Are you talking about the in vitro express or actually in the?
in the cell.
in the cell.
In basically in microorganism.
So some competitors actually wrote up a paper, and it came out early this year in Science Advances, and they did exactly just this, where they had figured out the centrin, I forget the name of the protein they identified, and they did a knockout of that thing. And the cell effectively can't contract anymore, but it can't, for some reason, there is some effect where it wasn't even able to maintain its shape. It basically looks like a crumpled cell. So there are some effects that they've explored in knockouts that.
Serious things, huh? Thank you.
Yeah.
Well, Saad, this was really inspiring. I think I certainly feel better about things right now. So that's wonderful. Thank you so much.
Thank you.
Keep up the great work and let us know how things are going.
Thank you.
Thank you.